All right. Hi, everyone. Good morning. I hope everyone's having a great morning after yesterday's wonderful dinner. Before I start our talk, I just wanted to start out by thanking the organizers for the wonderful summit that we've had so far, as well as giving us the opportunity to speak today. So my name is Juliet Frederiksen, and I'm a bioinformatician, as well as a bioinformatics manager at ZS Discovery. And I have the pleasure of sharing our talk today with Felipe, one of the bioinformaticians at ZS Discovery. In the first part of our talk, I will be introducing you to ZS Discovery, what we do, as well as the type of projects that we offer our clients. And then Felipe will follow up in the second part of our talk and actually walk you through one of these client projects where we worked to customize the NF Core single-cell RNA-seq pipeline to fit our clients' research goals. All right, so let's get started. So at ZS Discovery, we work closely with our clients within the early part of R&D to work, and these clients include, for example, pharmaceutical companies as well as biotech companies, and we work with their R&D departments to enhance and accelerate the R&D successes as well as capabilities in areas such as target discovery and data and knowledge management. And just as we at ZS Discovery work closely with the early part of R&D, we have other practice areas that work with other parts of the drug discovery development pipeline, such as medical affairs, as well as clinical development. And at clinical development, what they do is they work with clients to be able to enhance their clinical trial capabilities, both with design as well as operations. And now if we take an even broader look at ZS, ZS is a global services firm that is made up of over 12,000 individuals spread across 35 offices around the world. And over the last 40 years, we've become an end-to-end solution provider for both the life science as well as the healthcare industry. All right, so now back to ZS Discovery, which is where the two of us are located. And as I mentioned early on, we work with the early part of R&D, and we offer our clients these 4 different types of projects. And we have an especially proven track record for target discovery, as well as data and knowledge management. So for target discovery, we know that to find novel drug targets, we leverage advancements in both science, data, and technology to be able to find the targets that are supported not only in a biological systems level, but also with disease biology, because this will increase the probability of a successful drug discovery project. And now, if we move on to our data and knowledge management offering, which is actually really pertinent and very relevant for the summit that we're at today. This is where we work with data to collect it, analyze it, synthesize it, and present all of the relevant and key information. And in order to be able to do that, because we are biologists as well as bioinformaticians, we're able to work closely with our clients to get a good understanding of what their actual end goal is, and then help them design the process to get there as well as implement it. And this includes figuring out what kind of data it is, collecting it. This could be their novel internal data sets, but also going out into the rich public sphere to find the relevant data to fit the problem at hand. And so once we have collected the data, we then work to design and implement automated pipelines to clean up, analyze, and get the key information from the data and present it in both an intuitive fashion, but also a standardized way in things such as with Shiny apps, so that our clients' own scientists are able to dig through the data themselves to be able to develop as well as validate their own hypotheses. And then the other two offerings that we have are precision medicine. So this is very relevant in today's time and age, where we work to find the right treatment for the right patient at the right time. And finally, the last offering that we have is also the research strategy and transformation. All right. So to be able to help our clients face the challenges that they face, as well as deliver on all of the projects that I have presented to you, we span a range of different capabilities, as you can see here, as well as expertise. We employ various apps, and we're also able to analyze, but also understand various data sets and integrate them. And the only way we're able to do that is because we have a team of over 110 individuals. So we're an interdisciplinary team, and we like to call ourselves a molecular natives, because we truly want to understand the data that we are working with, not only from a technological point of view, but also the biology captured in there. But our team spans PhDs, medical doctors, bioinformaticians, computational scientists, statisticians, machine learning experts, and so on, and also people with wet lab experience that all work together on various different client projects. Additionally, we still routinely publish in peer-reviewed articles, and we do that not only through client collaborations and as a result of client work, but also because we like to stay connected to the academic ecosystem, and we still have a number of different partners that we work with in academia. And not only does this help us stay in touch with what's happening in the research world, but also helps us stay innovative for our clients. And so now we're coming to the end of the first part of the talk, and this is when I'll start wrapping up. So to finally just repeat, the types of clients that we work with are pharmaceutical companies, which range from large pharma to mid-sized pharma, but also biotech companies. And in order to make our clients' lives easier, as well as our own, we have come into a range of different partnerships. And one of them, which is very pertinent for, again, the summit that we're at today, is Seqera Labs, which through having this partnership, we're able to deliver more value to our clients. We're able to more seamlessly integrate solutions for them to get things up and running faster. And this is where Felipe will take over, and we'll show you an example of how this has happened. All right, Felipe, over to you.
Hello, everyone. Thank you for being here this morning after some party. I hope you're all pumped for today. Today what I'm trying to give us a brief showcase to you is like how the words of open source and, let's say, closed source can collaborate between each other, in a synergic way. So going to just showcase what happened in this collaboration with Boehringer Ingelheim that we had, where we needed to use, they needed to have something to analyze the single-cell RNA-seq data. And for that, there was already something inside the client's mind as well. They needed and they wanted to use the single-cell RNA-seq NF-core. I don't think I should give more reasons to because it's a gigantic community of people using it and making these things follow all the best practice analysis you can do for single cell. So it just makes sense to use this pipeline for the data, for the analysis in the client, and not reinvent the wheel if things are already there. But although the pipeline already provides all this core analysis that we need for the single cell RNA-seq data, the thing is, which is just ground true, institutions will never be the same. Each one has its own reality. Each one has its own set of data or type of, let's say, layered database that they need to communicate with. And this was the reality, for example, in BI. So this is like these three things. Is just trying to highlight how things were done and why the collaboration was needed. Because they were already using the Single-Cell RNA-seq pipeline. It was giving the results they want. They were pretty happy with that. But the thing is, they needed, because all the data, they need to follow data protection rules. They cannot have things in public databases or like stored in places where the NF-core pipeline is already like standardized to use it. So they had like a set of scripts, for example, which was the upstream processes that they had to communicate with this internal database of theirs. So they could fetch the data, store somewhere like in a bucket, because yeah, depending on where your data is, sometimes the data cannot leave that country. So all these first processings to let's say have a sample sheet ready to run the pipeline. So then once you have the sample sheet, of course, the pipeline was triggered, and then once the results were there, there was also some downstream processes that needed to be done, like because just having the data, just having the matrices was not sufficient for them, because who needs to see these results as the physicians is like the actual people that need to take care of these data, like need to interpret it. So in the end, there was still like some other scripts to make to work with these matrices, like to have some summaries, some statistics summaries that were relevant for the client, and then to finally upload it back to their internal dashboard where the physicians, where the people that need to see the data can actually see. Because giving them the files, it means nothing. They need to have that on the dashboard. And that's like something that they already had. But the things that they had that in a set of scripts, which, yeah, they were doing these things in 3 steps. And I mean, this is great. This was working for them. The main problem with that's like because you have these steps of manual work, it's not easy to scale that up. It's not easy to productionize that, and that's where the collaboration came from, because the result, like what was actually needed, is that we needed to internalize these manual steps as a single thing in the pipeline. So then you can have a single thing, a single execution pipeline that then it can be scaled up easily. So this was the internal, this is, let's say, the main goal of the collaboration. This is what was needed outside the collaboration. This is what was given to the client. So in the end, we had what we need to do, like this is, let's say, the major step or the major barrier, the hindrance that we had to overcome first was like, how can we internalize the single-cell pipeline? How can we internalize this NF-core pipeline? But in a way that we can customize some stuff that does not make sense to go to the public, because, for example, it's like just for these institutions. But it still be able to fetch to whenever a new update from NF-core was released, we could like get these new changes and update the pipeline internally without conflicts. And the actual answer is we'll never have not have conflicts. We will always have conflicts on merging them, but the thing is that you can make them less. You can make them like very minimal. So that was the first step. We realized like we could do this internalization, like allowing Git unrelated Git histories merging, and that happened. So on the way, we had this final project, this final pipeline that could do all those manual steps in a single execution. Like now, things are actually they are ready for the Seqera platform, which was so-called NF Tower. That's why you see that in the slides. But yeah, and now the actual way of executing it, it's way easier for the client, because instead of all those menu steps, they can just go to the pipeline and say, like, I want to analyze study 100. Then it knows how to figure that out. But you'll see that I'm not showing any of those things to you because that's not the actual goal of this talk. This talk, the actual goal of that is to try to talk to pump up the open source community, the wonders of Nextflow and NF-Core itself, because I mean all these was only able to happen because of the first, that the pipeline is in DSL2 and that DSL2 was integrated. I mean, I need to highlight that because even though that is not something very new, you'll still be amazed if you try to Google like how many DSL1 pipelines are still out there. And this just makes sense because sometimes you just don't have the resources to do the conversion, which is not seamlessly done. And then, of course, the NF-Core community. I mean, the NF-Core community, by adopting the DSL2 in most of the pipelines and also making these collaborative communities, this is what actually make all these things happen. But then, so far, I've been telling, like, okay, this is how we benefit from the community, but what does the community get with that? Like, so that's the actual endpoint where we want to highlight that, like, this is not supposed to be a one-way thing. It's not like just a closed source that should use what is open and that's it. It should be a synergic, a mutualistic association between these both communities, let's say. And this is what we wanted to integrate. Like if you see today the collaborative history, like the commit history of the pipeline itself, of the repo, following all the BIs like needs, all the specifications, everything that they wanted. Like we did a lot of, also pushed back a lot of commits to the community because, for example, when doing the analysis, we are also facing some stuff like, we need this thing. Let's say we need this matrix conversion. This is one thing that we added there, for example, to CRETs, to H5AD or whatever to use with other tools. And then we saw that in the GitHub, in the public repo, these were also an issue open there. So I mean, instead of just adding in our internal copy, we are like actually adding these modules in NF-Core modules, then pushing them back into the single cell, because I mean, we could separate what changes we wanted could be public and what changes we wanted should not be public. But then, like, yeah, this is the actual main source, and I think the goal of the whole talk is like, I think that closed source communities, companies, or the private world, the pharma world, can benefit from it, but also I think by this happening, the open source community can also gain a lot. Yeah, and I would like to finish it like by thanking everyone, and because like to put names in the slides would not fit in just one slide, we decided to just put the names of the organizations, and I want to thank everyone for making all these happen. Thank you.
Thank you so much. That was really interesting talk. It's a really interesting model for kind of driving innovation and improvements. We have one question for you from Slack. Was your client ever hesitant about you putting work into an open source project?
Nope.
No. Wonderful, excellent. Wonderful clients. Another question for you. How much customization was required to get the pipeline working with Tower, which is now Seqera platform? Was this simply standardization to NF-Core scheme?
Yes, I mean, there was actually no trouble at all. I mean, because the pipeline, the NF-Core pipelines, they are already ready for Seqera platform. By using the schema and all, I mean, whatever we added for the private world, let's say, like whatever we added for this internal database thing, and we needed new parameters or whatever, we just needed to follow the same standard. But I mean, there was no issue. Yeah.
Got it. There are more people typing in the Slack, but we'll thank you for now. And if you'd like to continue the conversation, I think there's a lot of interest in the model. model.
Yes.
So thank you very much for your talk once again.
Thank you.